what I'm going to talk about first is describe what a tracer is, talk about the perfluorocarbons, which we call PFTs, so I'm going to say PFT when I mean perfluorocarbon tracer. Why they're good tracer compounds, how we use them, some of the things we've used them for and what we've learned when we've done it, and some of the things we'd like to do with the PFTs in the future. Now, why they're important, we can address all kinds of problems of mass transfer. Air quality is a good one right away. You want to know where the mass, the pollution that's coming out of a coal-fired power plant is being transported, whether this coal-fired power plant is in China or whether it's in the Tennessee Valley and how it's going to affect the Northeast. It's also important to identify mass transfer processes in climate studies. How is the CO2 cycling in the atmosphere between the air and the terrestrial biosphere? So we want to try to identify how this mass is moving in the atmosphere and between biological systems in the atmosphere. So there's a mass transfer. It certainly is important in terms of toxic releases. This is the Fukushima plant after the tsunami disaster, and you really want to know where the toxic cloud from that release from that plant is going to go. Emergency responders are particularly interested in mass transfer questions. They want to know who has been exposed to various toxins that have been released, where they need to do the cleanup, and how they're going to approach these sites where there's been an accident in order for them to deal with it. It also has important implications in homeland security. This is the aftermath of the Tokyo sarin releases in the Tokyo subway, the Madrid bombing, and the London bombing. So the emergency responders are particularly interested in how to address these types of issues, and they want to know where things are, how they're being moved through the atmosphere. Building ventilation is another thing that we work with and do with the tracer tool. In a nuclear power plant, you have a control room where the operators are working. They have specialized ventilation systems that will eliminate any radioactive material that's during an accident when there's a release, so the operators can work in the plant safely and mitigate the problem. So we test those systems using our tracer technology to make sure they work well. We've done some work in Building 400 here on site, which I'll talk about a little later. We've done some work in the biggest, actually a lot of extensive work in the biggest office building in the world. And we've done some work in some office buildings in New York City where we're actually trying to figure out why there's a smell from the Chinese restaurant that's next door that's showing up in the offices in the lobby of the building. And we actually put some tracer material in the exhaust hood of this restaurant and found it in the buildings. And what we learned is that the exhaust from the fume hood was pretty close to the intake for the air handling system. And finally, we use these in underground systems, like toxic waste containment structures. You want to make sure that the waste is staying in and not getting out. Underground electrical cables, natural gas pipelines, and other underground systems so that we can identify leaks quickly and take care of it, which has obvious public safety implications. So what is a tracer? A tracer is an indicator that's added to a system so the movement of another mass, another substance, can be tracked easily. There's a lot of systems like, for example, a natural gas pipeline. If the natural gas is coming out, there's a lot of natural gas in a lot of different places. You want to find where the leak is, so you put a tracer in there so you can pinpoint it exactly. One of the earliest uses of the word tracer is for the specialized machine gun bullets that they put in a belt of ammunition that's going through a machine gun. Every fifth shell or so has some phosphorus in it so that when it's fired, that it ignites and it releases a cloud of gas or a stream of light. So here's a tank, which is a little hard to see, I guess, and it's firing these bullets out, and every fifth bullet or so glows, so you can trace the movement of the mass, the bullets, to the target. I kind of wonder if that's a good idea, because you kind of trace them back, too, to where they're coming from, but I don't know. Yeah. So the tracer characteristics, there are two important characteristics you have to make a good tracer. The most important one, it has little or no background level. And second is that you can easily detect this material at the background level. So what is a background level? Background level is the basic level of something in the environment that you're talking about. For example, if you're in a crowded room and there's a lot of conversations going on, there's a background level of conversation. If you want to hear somebody speaking to you, they have to be close to you and speak fairly loudly, so you or if they're far away, they have to raise their voice above that background level. Now, in the atmosphere, the air is composed of a lot of different substances, mostly nitrogen and oxygen, and it has 1% of trace species, which is where the part that we're really interested in is that 1%. Interesting that carbon dioxide in the atmosphere as of last month was at 393 parts per million. I remember when I started paying attention to carbon dioxide levels in the atmosphere, it was about 370 parts per million. I guess this is kind of like, my dad used to tell me that gas was 30 cents a gallon. So back in the distant past, we actually had different levels of CO2. So I said parts per million, that's a mixing ratio. I want to describe what that means. A percent is a mixing ratio as well. A percent is a part per hundred. So if you have 1%, you have 1 part per 100. We're talking about parts per million in most of the atmospheric contexts. So what does that mean? It's an expression of concentration in terms of the ratio of the number of molecules of one compound to the total number of molecules in the sample. We also call this, in chemical terms, a molar ratio. It's independent of sample size, which is why it's so useful. And it's independent of altitude because it's independent of sample size. At sea level here and at 0 degrees C, there's about 2.4 times 10 to the 25th molecules in a cubic meter of air. A part per million is 1 molecule of the compound in 1 million molecules of air. So if you had a part per million in a cubic meter of air, you have about 10 to the 19th molecules in 10 to the 25th molecules in that cubic meter. So if you subtract 25 from 19, you get 6. 10 to the 6 is 1 million. Okay, one of the best examples of a tracer that everybody's familiar with is natural gas. Natural gas, as it comes out of the ground at wherever they're producing it, is odorless. What they do is add a substance called mercaptan to the gas. This gives it that distinctive odor that when you smell it, you say, that's a gas. So it's obvious. That's a tracer. The mercaptan tells you that it's something that indicates that the bigger mass that you're interested, the gas is coming out. The smell of it is distinctive and unpleasant, and it's distinctive is important. That's like it means it has a low background level. You don't smell mercaptan anywhere. It wouldn't do any good if they put something that smelled like bacon, in the gas, because you smell bacon and you think there's a gas leak. So it's got to be distinctive. Okay, and the interesting thing too about mercaptan is the human nose is incredibly sensitive to this. It can see it at 10 parts per billion. So there's that mixing ratio stuff again. Yeah, 10 parts per billion is pretty low. Okay, to the perfluorocarbon tracers. This is our tool, this is our hammer. And just like what tools that you use for any particular job, we can scale our tracers to the job that's necessary. If we have a really delicate job, here we have a hammer. This is a reflex hammer. So what the doctor uses to hit your knee when he tests you. So that's something that we use in some instances where we have to be very careful what we do. When we're looking at brute force, we can use a sledgehammer. And when looking at finesse, we can use a finish hammer. So we can scale the way we use our tracers to lots of different applications. So what are these things, perfluorocarbon compounds? The PFT technique, perfluorocarbon technique, was developed here at Brookhaven by Russ Dietz back in the early 1980s. And he specifically worked on this to apply to atmospheric transport and dispersion research. The whole genesis of the tracer of atmospheric tracing goes back to atomic testing and the development of nuclear power plants. They wanted to try to find some ways to see how things were dispersing in the atmosphere, and the Department of Energy and NOAA actually developed perfluorocarbon tracers based on the input from James Lovelock, who developed an instrument that works real well with these materials, to come up with ways so that they could see what's happened when there were nuclear testing, or what would happen if there was a release from a nuclear power plant. And the nice thing about our tracers, we have 6 available compounds, which every time we take a sample, we basically have 6 data points that we can use. So that amplifies our ability to do testing. The perfluorocarbon tracers are present in the atmosphere at very low background levels, from less than 1 to approximately 9 parts per quadrillion. It's a mixing ratio. I'll talk a little bit about more in a minute. And they're detectable at the background levels using the specialized gas chromatography methods that Russ Dietz has developed, which are fairly complex and work really very well. So what is a part per quadrillion, a ppq? This is actually, it was really fun for me to think about this and to try to come up with a way to describe it, because, you know, I say ppq and part per quadrillion all the time, but I never really tried to think about what it was, and other than just some concept in my mind. So I had to sit down and come up with some way to explain it to put this talk together. And first thing you say is it's 1 molecule of PFT and 10 to the 15th molecules of air, or a mixing ratio of 1 over 1 with 15 zeros after it. Well, that's not very satisfying. What does that really mean? Okay, so what I decided to do is say, if I had a gram of PFT, what volume of air would I have to mix that into in order for you to get a part per quadrillion? Okay. So this is a gram of PFT here. It's about half a cc. Okay. The volume of air you would need is 6.4 times 10 to the 10th cubic meters. That's 64 cubic kilometers. That's 1 kilometer deep over an area of 64 square kilometers. Another way to look at that, that's 6/10 of a mile deep over an area of 25 square miles, or 6/10 of a mile deep over an area equivalent to 15,000 acres, or about 3 times the area of the laboratory. So if you had a volume of air a little bit more than half a mile deep over an area the size of the laboratory, and you took 1 gram of PFT and mixed it into that volume, you'd have a part per quadrillion. Okay. I had fun with this, so I said that would also be about 31,650 city fields. I thought about using that stadium in the Bronx, but I figured out it was so full of hype that you couldn't talk about volume. Okay, one more. If you had a volume as high as the Empire State Building, it would cover, you'd need an area of 3 times the size of Manhattan in order to get a part per quadrillion. Okay. We can detect background levels at the part per quadrillion range if we take 1.5 liters of air. So you take that 1 gram, mix it into 64 cubic kilometers, then we can take 1.5 liters and we can see the material. Okay. The PFTs are chemically inert. They're non-flammable, no biological effects. There's actually a lot of medical applications that people have studied and used these for. They actually put them into microcapsules and inject them into people and use them as ultrasound contrast agents. And then the microcapsules dissolve, it goes into the bloodstream, and people exhale the PFT into the atmosphere. Okay. We call them by their acronyms, which we have developed. So these are, if I say PDCB, that means we're talking about a particular perfluorocarbon. The important point here is they're all carbons and fluorines, different numbers of carbons and fluorines, but no other elements in the compounds. So why do they make good tracers? First of all, the background level, the first characteristic of a good tracer, low background level. Background levels are on the order of less than 1 to about 9 parts per quadrillion. Okay, and this is data from a paper that was in Environmental Science and Technology back in 2007. Okay, The second criteria, we can detect them at or near the background levels. This is the limit of detection and limit of quantitation of the analytical method that we use. Basically, we determined the standard deviation from one of the lowest, using the lowest level of calibration standard that we use for each of the PFTs. And then, I consider that the background noise level, the level that we have to rise above in order to see a detectable signal. The limit of detection, which is the amount of signal we have to get to know that we see PFT that's above background level, which is 3 times the noise level. Here from 3 to about 9 parts per quadrillion. And then the limit of quantitation, which is the level we want to see if we want to have a 30% uncertainty level on the number that we get, is 10 times the background noise level, and that's here. Those are kind of arbitrary numbers, but they give you a good idea of how sensitive our detection methods are. The way we use these, basically tracer program has, you release the tracer in some way, you sample for it, and then you analyze. Okay. We have a number of different release methods that we can use. These can be active or passive, and we can use these to scale the release, how much PF2 we're using for the problem we're trying to address. Again, background levels are in parts per quadrillion level, and one of the drawbacks of using these things is with these kinds of detection limits, we have a problem. So you have to be very, very careful that you contaminate yourself. You know, when we're doing large-scale tracer releases, one thing we often do is have the people who are releasing the tracer kept completely separate from the people who are doing the sampling, because you can just by being around them, you can get maybe parts per million level in your breath. And a part per million level is a lot more than a part per quadrillion, and that you can contaminate samples that way. So you have to be very careful about how you deal with your materials. Okay. The sledgehammer, the brute force approach when we do a release. This is a spinning cup release system. It consists of a metering pump that puts PFT through a tube into this porous ceramic cup. This is a blow-up view of the cup. The PFT drips into the cup spins at 30,000 RPM, and it's porous, so the PFT is forced out through the pores in the cup and creates a vapor that disperses. We use this to release tracers at kilogram per hour levels, and we can, the experiments where we need that kind of release rates are hundreds to thousands of kilometers. And we have the longest distance that a PFT tracer atmospheric dispersion experiment has been, it's about 2,000 kilometers. Okay. Our little more finesse, where we use our finish hammer, is in the grams per hour range. And for this, we have a couple of systems. The first is a syringe pump. This is basically a metering pump that's used to administer IV drugs into, you put it in a syringe, and it compresses at a measured rate to introduce drugs into the IV in a hospital. So we use this to introduce the tracer onto a piece of filter paper with a fan in the box, and blows out, and we can use this on scales of kilometers to hundreds of kilometers. This is the type of release apparatus we used in New York City. We can also make up gas mixtures of the perfluorocarbon compounds in nitrogen and release those at different rates. We use this kind of system to release gas inside the Pentagon in bursts, because they were interested to see what would happen if somebody took a bottle of some toxic substance and smashed it. Okay. Now our finesse methods, the reflex hammer methods, are passive release. For this, we use a serum vial. There's a PFT in this vial, and then there's a permeable silicone membrane at the top of the vial. And the PFT in the headspace diffuses out through the permeable membrane and into the atmosphere. This is micrograms of PFT per minute. We use this a lot in indoor studies, building infiltration, air purification evaluation, and outdoors in some really specific but short-scale studies where we're talking about meters to hundreds of meters distance we're trying to cover. The amazing thing about this, if we were in this room and we took and put one of these vials in this room and let it sit for 24 hours, I could come in here and we could take a sample, then we'd have more than enough signal. Okay, and the sampling methods are also, we can scale to different applications. We collect the samples on activated charcoal. That actually concentrates the tracer a little bit, so that helps us with getting a good detection limits. One important thing is that all 6 of the compounds can be resolved in a single analysis. So that goes back to every time we take one sample, we can get 6 data points, which is really important. This gives us a way to address multiple issues when we do an experiment. We can put a bunch of tracers in a bunch of different places, so we can see, we can do an experiment where we talk about what happens if a tracer is released in the subway, and what happens if a tracer is released on the surface at the same time. So it gives us a lot more flexibility. The other thing it gives us a lot of flexibility in doing is quality assurance and quality control. Sometimes we will take and release 2 tracers at the same location. So then we better see the 2 tracers at the same levels in our samples, and that always happens, so we can use that to verify that our sampling methods are working well. Okay, the sampling can also be either active or passive. Okay, these are the charcoal, are the absorbent tubes. There's the charcoal absorbent bends. This is a sealed sample, and this sample is, this open tube is actually sampling. And I have a tube here, so if I take and just take this cap off the end of the tube, like that, okay, and hold it up, now this tube is sampling. It's by diffusion. And the amazing thing to me is that the rate of sampling is about 0.2 of a liter per day, which I was surprised when I calculated that. Okay, and we take these tubes and do leak hunts for underground dielectric cables routinely. We'll take in the underground cables have dielectric fluid and oil that flows through the cable to help cool it. It's not like when they have a suspension cable, it's suspended in the air. If it's underground, you have to do something for these high voltage cables. So there's an oil in there, and they leak. Then the utility company has to find the leak. So what we will do is tag the dielectric fluid in the oil, and then we'll take a bunch of these tubes, and usually these are along roads, and the cables run along roads, and we'll go and put these samplers on telephone poles, leave them out for a week to a month, come back and analyze them. You take a look at them, and you can see nothing, 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 a little bit there, lots, nothing, 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 as you go along, and you can pinpoint the leak. Once we've done that, we say it's in between this telephone pole and that telephone pole, and we go back, we go look in the ground, and we can pinpoint it within a couple of feet. Okay. We also have active sampling. This gives us time resolution. We want to get about 1.5 liters of sample onto our charcoal absorbent. So we actually pull air through this charcoal absorbent. Here's the tubes inside this particular sampler. It samples at 500 mils a minute, so we can get our 1.5 liters in 3 minutes. We can actually do a little bit better than that in terms of speed, so we can get pretty good time resolution. Here's another pumping system. That's a quarter right there. This is a personal air sampler. It's about this size, and we've done experiments where we take and put a tube on this, and people walk around in places where we've released tracers, and then we can see what their personal exposure to the tracer was, and use that to extrapolate to what would happen if it was some other kind of substance that was being released. Okay, the analytical method, the third part of doing a tracer experiment, is done by gas chromatography with electron capture, or ECD. It has a sensitivity, again, to get the background levels, to get parts per quadrillion levels if we have 1.5 liters of sample. Now, in the atmosphere, there's interferences. If we didn't eliminate these, they would come interfere with the signal from the PFT. So this is the magic of Russ Dietz, is to go in here and create this chromatographic method, which has dual columns and clean up stuff, and he waves his hands over it every once in a while, and it's a system that is pretty unique. There's not that many people in the world who can do this kind of analysis. Okay, this is an example of what comes out of our instrument, the raw signal that we get. We have millivolts here, which is the response of the detector, and time here. This is about a 10-minute time scale down here. And you can see the colors aren't coming out here the way they did on my laptop, but this is one PFT. That's 2, 3, 4, 5, 6 here. The reason that there are a couple of different peaks here is because this chromatographic method actually can resolve the isomers. What a chromatograph does is you've got a lump of material, and you put it in this machine, and it travels through the machine, and as it comes out the other end, that lump of material, the different components have been strung out and spread apart. Okay, that's why it takes 9 minutes for this to come out every time, and when the time that it comes out is unique to the compound. So we have a bunch of compounds. This compound is the same chemical formula, but it has a different physical structure. And that gives us some internal standards. We can look at the ratios of these isomers to each other, and it can give us another way to check our quality assurance and quality control. And we can actually even use the atmosphere and the composition of the atmosphere as an internal standard. This one is what the new equipment upgrades that we've made, and so for this, instead of a hammer, I put a nail gun on there. Okay, we've got a new analytical system with LabVIEW, and it's just wonderful. Does a lot of different stuff. And right now, we're in the process of building 100 new tracer samplers that are the samplers we're using now were built in the 80s, and now we've got a whole new set that's coming through the pipeline and with all kinds of bells and whistles. One of the really nice things about these new samplers are they have Wi-Fi in them, so you can go around with your iPad or your iPhone or whatever, and you can see what's going on with the sampler. If you stand there, you can see it's running. When we did the urban dispersion program back in 2005, Rick Wilkie and I went around with a stethoscope, and we would get up and we'd put the stethoscope on the sampler and listen for the pump and say, oh, that one's running. Excuse me. Okay, so the nails, what do we do? How do we use these PFTs? I want to talk about long-range transport first. This is the BRAVO experiment. It stands for the Big Bend Regional Aerosol Visibility and Observational Study. One thing that's obvious from calling this thing BRAVO is that acronyms are kind of silly. These people come up with the acronym. We did one of the Pentagon programs was called PANDA, and that was another one. They just came up with a name and tried to figure out an acronym that it would fit. Big Bend is a national park right here at the bend in the Rio Grande, which forms the border between Texas and Mexico. And the Big Bend Park is right here at this place. And Big Bend has some visibility problems. This is an average low haze day. You can see off in the distance the different mountain ranges. It looks pretty interesting. But there's a problem from sulfate aerosol, which is produced by power plants, coal-fired power plants primarily. So that when there are hazy days like this one, the sulfate aerosol is in the park and it reduces the visibility. And this is particularly important and to the people that go to national parks because it really wrecks your experience if you can't see anything. So the National Park Service is doing a lot to try to find out what's causing visibility problems in the national parks. The objective of this study was two things: to determine transported dispersion from power plants. They actually wanted to see what specific power plants, how the material was transported from them to Big Bend, and also to evaluate and improve the performance of air quality models. And this is something, this evaluate and improve performance of air quality models is something that comes up in just about every one of the atmospheric transport and dispersion experiments that we do. We're always trying to collect data sets that they can use to try to evaluate their models and try to figure out where the problems are so they can make them better. The fact that we can get some empirical data actually is very important because about the performance of the models, which I'll get to. Okay, here's the setup. We have these release points, the Big Bend, the Brown power plant here. We actually released at Big Brown and Parish, our two power plants, we actually released perfluorocarbon right into the stack. We ran along a line up the side of the stack and injected about 100 feet up into the exhaust stream as it was coming up through the power plant. We also released, Big Brown is, 725 kilometers away from Big Bend. This is the line of samplers that we put out. There's one right here at Big Bend. Parrish is this from here to here. It's about 850 kilometers. We released at the surface in San Antonio, 450 kilometers away. And we released at Eagle Pass, Texas, from a television tower about 350 feet above the surface. The reason we released at Eagle Pass, Texas, was because there's about 32 kilometers away, there's some power plants in Mexico, the Carbone 1 and Carbone 2 power plant. And the Mexican government wouldn't let us tag their power plants, so we got as close as we could, and did a release to try to simulate what was coming out of the power plants in Mexico. Okay. Here's some of the signals. This is from the 850 kilometer away power plant, the Parrish power plant. So we have the tracer concentration on the y-axis and time in days, and this time spans 1.5 months. And these legend indicates the different sampling locations. What's clear is that we can see a definite signal from the power plant 850 kilometers away at the samplers that were in the Big Bend region. Also, this is another one from the Big Brown power plant, which is 750 kilometers. So we get good signals over that great of a distance. Okay. First, the results of this project I want to first address the modeling. The details here aren't important, all these acronyms, but there's a bunch of different models that they tested in order to see if they could simulate the tracer releases. And what happened was that none of the models did very well. This is right here, this black line with the error bars on it is the actual data from the tracer release here. And the red and the blue lines are the data from the modeling. They took the release information from the actual releases that we performed and put those as inputs into the model, and then tried to see if they could predict what the tracer concentrations at the receptors were. So you can see that they didn't do a very good job of predicting. In fact, it's almost random when they predict lots of tracer. There's not very much, and when we see lots of tracer, maybe some agreement, but not really. So, and correlation coefficients are really bad in. If a good correlation coefficient would be 1, an acceptable correlation coefficient might be 0.9, and these are very, very low. The difference between the blue and the red is the way they initialize the models. They take different ways to input wind fields into them, and some work better than others. And it's peculiar sometimes. One way of doing it works better than the other way of doing it, and there's no real clear pattern. But we also had empirical data because we did an experiment, we knew what we released, and we saw stuff, and we knew where we saw it. So we could actually get some information out of that was useful. One thing, we could calculate the transport time from Eagle Pass to Big Bend pretty exactly, 15 to 18 hours. Also, we determined dispersion factors because we knew what the release rate was at Eagle Pass. We knew how much we saw at Big Bend. We can say, okay, it gets diluted this much from here to there. So the Carbone power plants will get diluted somewhere similar to that. So we could actually get an idea of what was happening with transfer from the plants in Mexico. So 4% on the least haziest days, 8% on the haziest days was coming from Mexico. So the Mexican power plants weren't contributing a lot to the haze. What we determined was the SO2 sources, the power plants in Texas, and also the power plants in the East Coast were the biggest culprits in contributing to the sulfate aerosol at the Big Bend National Park. Just a list of the publications that came out of that. When I move on to the urban dispersion program in New York City, this is a really nice pictures from where we did a lot of our planning at the Stevens Institute in Hoboken. The conference room has this view. It's just, I can't resist showing that. And right over around here is where we did, that's a Penn 1 building right there, where we did the Madison Square Garden study, which is what I'm going to talk about. The reason that urban dispersion is important is because that buildings have a big effect on how material disperses in the atmosphere. This is a study of smoke releases that simulate what happens here. There's no obstacles in the way. You can see the plume is very narrow and fairly regular. Okay, here there's some blocks simulating the urban canyons, and that the dispersion is much greater. Okay. Objectives of UDP: to provide a data set for model development evaluation again. Okay. Practical guidance for emergency responders and identify gaps in the understanding. When the models don't agree with the empirical data, there's something in the way we're formulating the models that we have to look at. So that's the gap in our understanding. This is the setup. This is approximately north. This is Madison Square Garden here. This is the Penn 1 building here. We released at the four corners of Madison Square Garden and also on the opposite side of Penn 1 from Madison Square Garden. So there's 4 tracer releases. Okay. The first one, the experiment that we did, the mean winds were coming pretty much parallel to these street canyons here across the rooftops over Madison Square Garden with our sampling here, 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 and on the other side of Penn 1. Okay. This is a diagram of the wind fields. We've had meteorological instruments on the roofs of the building around the Madison Square Garden and also on the streets where we did the releases. The roof of the buildings are indicated here. This is just to show you that 5 meter per second wind speed is that length of a vector. So this is about a 5 meter per second wind speed here. The cone around it indicates the uncertainty, the variability in the winds. So the winds were pretty much in this direction, varying a little bit here at the rooftops. But you go down and look at the winds at the surface, and you can see here they're perpendicular to the direction of the wind at the rooftops, which is sort of what you would expect if the wind came across here, ran into this big building. Remember, here, wind comes across, runs into this building, gets deflected in all different kinds of ways. Okay? And here it's in the opposite direction. Okay, over here on this other side of Penn 1, it's in this direction with this kind of uncertainty. And here at this corner, there's no arrow because there was the wind varied about in all kinds of different ways, so there was no real resultant. So the variability was all over the place. Okay, so from this, we got plumes from the different tracer releases. This is on the north corner of Penn 1. Here, you can see the tracer actually traveled upwind and perpendicular before it started to disperse in the downwind direction. Same thing, this corner here, we're going over here to this corner of Penn 1, traveled in the upwind direction before it began to disperse. On the other side of Penn 1, where there's no building in the way to block and deflect the air, pretty uniform. Over here on this corner, from here we go to this corner, down here, okay, back up in the upwind direction as well as downwind. This corner here, from here to this corner, over here. You see it in both directions in the upwind direction. That's just the diagram of the winds again. Okay. So from this, we were able to determine some practical empirical guidance to give to emergency responders. The first is that if there's a release at a point here, you want to stay a half a kilometer upwind of from the direction of winds indicated by the rooftops. The other is that the cone of exclusion extends like this, about 2.5 kilometers from the center line of the plume. This is a lot larger than the safe zone that you would determine if you just used a straight Gaussian plume model. The plume is much smaller, so we've been able to say that you ought to this is the zone where you'd have to evacuate and the zone where you don't want to go into. Okay. We take this and pose it on top of the plume diagrams I showed you earlier, and you can see how we fit it in. And here, there's an indication of the non-building awareness plume, that narrower plume that I showed you earlier. So we've been able to expand the exclusion zone from a non-building aware plume to this size. And see, you can see that it fits all the empirical data. So this is no models. This is just taking what we did in the experiment and using that to create some kind of guidance. I'm going to talk about what we did in Building 400. We did some scaling studies a year and a little more ago at Building 400 where we were trying to test the release rates we were going to use at the Pentagon. But while we were doing that, we decided that we ought to, some people were complaining about smoke inside the building. So we decided we ought to do a study to try to see what we could determine about smoke from the smoking areas outside the building getting into the building. So here we have Building 400. There's the Starbucks right over there. All the tables where everybody sits. Right over the street would be out here, and the library would be over here. So we released 6 tracers. We released one right in the atrium. Then we released one over here in the designated smoking area, one over here in the not designated smoking area where smoking occurs by the stairwell, one out here in this front courtyard, one here, and one right there. Then what we did is we normalized all the release rates, all the measurements to the release rates so that we can compare all the different PFTs, and we compared the level of the PFT that was released inside the building to the level of the PFTs that were released outside of the building. And what we found that in most cases that the tracers released here, 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 and here were present at about 1% the level of the tracer released inside the building. The tracer that was released here was present in the north sector of the building, at 8% the level of the tracer that was released here, and in this part of the building was really present at 3% of the tracer that was released inside the building. So if people are smelling smoke, that's where it was coming from. Okay, and that's what that says. Okay, not everything is a nail. We've used this in a lot of different applications, but not every application is appropriate. We just published a paper in Environmental Science and Technology that's electronically available now, and they haven't told me when it's going to be published in the hard copy issue. But where a lot of people are proposing that we use perfluorocarbon tracers to tag the CO2 that they're going to remove from power plant plumes and then inject into saline aquifers about 1 kilometer down underground. The variability of CO2 in the atmosphere is so great that they don't want to, that's really hard for you to measure directly measure CO2 and see if there's a leak. So they want to put tracer in the CO2, look for the tracer, and see if there was a leak. The problem is that it's going to take just too much tracer. And economically, it doesn't make any sense. And that kind of tracer, the amount of that, you would need more than the total atmospheric burden of tracer that exists right now just to tag one power plant. Okay. There are plenty of nails, though. Even though we can't always use tracer to do everything. What are some of the things we like to do with PFTs in the future? We're working right now with microencapsulated PFTs. These are actually little shells that are filled with PFT liquid, like a ping pong ball with PFT liquid inside of them. The scale, here's 10 micron scale here, and these are all different scales. Maybe that's 50 microns, maybe these are 8 or 10 microns. What we want to do with this is to use them in different systems. Well, if we can get the PFT into these little, and we actually have microbeads. This is a scanning electron microscope of some actual PFT in microbeads. We want to use them in different systems. We want to use them in aqueous systems because PFTs are insoluble in water, and they're also soluble in hydrocarbons. So if we want to trace a leak from a water main, for example, as soon as the PFTs, if we put it in the water directly, as soon as you get headspace, the PFT's gone. But if we put them in the microcapsules, we make the microcapsules small enough, like a micron or so in diameter, they won't gravitationally settle. We can take a sample of the water, dissolve the microcapsules, measure the PFT, and we can use them in aqueous systems that way. They're also soluble in hydrocarbons, which if you're trying to do stuff in oil wells or in where there's any kind of hydrocarbon like coal, you don't exactly know what's going to happen to them because there's a lot of different things that could happen when they go. So they'll go into solution in the hydrocarbons. So microcapsules can help us address both of these problems. I just love this picture. This is a fracking experiment in West Virginia. These are tanker trucks of liquid nitrogen. They're taking this that much liquid nitrogen and pumping it down into into 5 different wells in order to break up the shale so that they can get the natural gas out of it. Before I saw this picture, they asked me if we could do some stuff to help them. I'll show you in a second. And so I gave them a number, a couple $100,000 to do this. We had to figure out what was going on with the coal and the PFTs and stuff. It was a research project more than an actual application. And then they said, no, that's too much money. And then I saw this and I said, boy, I should ask them for more. Okay. So this is the application. They have a bunch of different wells, and they drill them down and then go horizontally. What they wanted to do was, as they were injecting the nitrogen, was to put one PFT in this leg, one PFT in this leg, one PFT in this leg, and one in this one. And then after they put it down and let the liquid expand into a gas and do the fracturing, they want to see when the gas comes back out if the PFT that was put into this leg comes out into gas here, so see if they got interconnectivity between these different legs, because they don't know. Okay. We also, geothermal well characterization is one of the other microcapsule applications. Simulation of particulate releases, which is important in the atmosphere because we release these PFTs in the gas phase, but how do particulates behave? So if we could have PFTs and microcapsules that we could release into the atmosphere and collect them, then we could compare gas phase to particulate phase releases and see how the particulate transfer is working and verify some of the parameterizations for gravitational settling that are used for particulates. And we can also use them in dispersed release systems. There's some talk about actually putting these into polyurethane spheres, putting them in latex paint, and painting something with it so you have a really long, big, huge release area. Future projects, 2012 and 2013, we're going to be back in New York City doing some work, transport and dispersion. There's a possibility, I don't know how good it is, that we'll be in Indianapolis in the fall of 2012 doing some CO2 flux experiments where they're trying to measure CO2 coming out of Indianapolis from various anthropogenic sources. And one thing that I'm really I've been thinking about a lot and I think is important is that there's a need for a lot more long-range atmospheric transport and dispersion tracer experiments, because the Bravo was the last large atmospheric transport experiment that was done. It was back in 1999, and really large, long-scale programs, there hasn't been a good one since the 1980s. And these are the data sets that most of the modelers are using to get their parameterizations now. We have a lot better sampling. We have a lot more samplers. We can give a lot better resolution. And I think it would be a really good idea to do some more long-range transport and dispersion experiments. So I'd like to conclude by thanking the people that support the Tracer Technology Group. Russ Dietz, who's now retired and is in Florida, and I couldn't get him to come back to be here. Rick Wilkie, who runs our analytical systems and takes care of all the instruments. Gabe Vignato, who's builds everything for us and just did a really nice job of putting the plumbing in our new lab. Anybody wants to see a nice job, come and we'll show them to you. And auxiliaries, Paul Cab, who happens to be the boss, John Heiser, Scott Smith, Larry McMillian, and Andrew McMahon, who are not exactly in the Tracer group, but are sort of like half in the Tracer group and half out. We use them when we need them, and it's a real pleasure to work with these people. I'm really, I've said it before, and people know it. This is the best job I've ever had. So thank you very much. So I'll try to answer any questions if anybody has any. Steve?
Tom, when you look at the park, the Big Bend,
yeah.
It seems that most of where you released it was east of the...
Yes.
And I thought the prevailing winds they go from west to east. So
yeah,
how come it goes backwards?
It depends on where the the system is located.
You said there's contamination from the East Coast.
Yes, and oh, here we go. Here. If you've got, the circulation around the low is counterclockwise and around the high is clockwise, right? So if you've got a low system down here traveling this direction, you're going to get this counterclockwise circulation, and so the air's going to actually move in this direction on the top side of the low. See, I'm a chemist. I'm not a meteorologist, and I can say high and low and Pascal Gifford stability class, but I don't know what they mean. Anybody else? Okay. Okay.
Yes. My question, basically, you touched upon industrial applications for tracer gas, building ventilation, overhead to floor is often used for that, as well as energy efficiency.
Yes. Yes.
One of the constraints that I'm seeing now is that because it's a greenhouse,
Yes. Yes.
this is also a greenhouse.
Yes.
And I guess comments on, first of all, is it viable for these kinds of also is it economical?
Really good question. And sulfur hexafluoride background levels are in about 7 or 8 parts per trillion, right? So that's 3 orders of magnitude higher than the PFT background level, just to start with, okay? They're also increasing because sulfur hexafluoride is used a lot in electrical switchgear as a spark inhibitor. So there's a constant release of sulfur hexafluoride over the globe. So when I first started doing this back in the 90s, I was doing sulfur hexafluoride, and the background level was about 2 or 3 parts per trillion, and now it's up around 7 or 8. So you need 1,000 times less PFT to get a signal than you do sulfur hexafluoride. Okay, greenhouse potentials are probably about the same, but when we did a lot of our dispersion studies, we release grams, I mean like less than 100 grams, to do the six different tests of 6 different PFTs. We don't release, the amount that we release is insignificant. You can't, the global background of the PFTs are, for all practical purpose, constant. Forever. They don't break down until they get to the mesosphere. They say, okay, the best estimates I've seen are 3,000 years, but that's kind of a guess. So yeah, that's definitely a drawback. But the way we get around that is just to say we release just a really little bit.
The next question, because the molecules are so small and so light, for emergency planning purposes, what's the release potential at the ground? Meaning, do they stay at ground level, or is it just an even distribution throughout the atmosphere?
They don't stay at ground level. There's enough turbulence that they mix up. We see them almost, if released at ground level, at the tops of buildings that are from 25 or 30 stories high to up to, I forget what Pen 1 is, but Pen 1's a big, how many? 80 stories. We see them up from a release from the ground, up at the top of the building quickly. Right.
Where a lot of the air intakes on buildings.
Right.
Is there a direct correlation between the wafting of the PFT and then toxic material that might have a vapor density much, much greater than air?
Right, and then some of these things are sticky, some of them are reactive, so and the PFTs are none of those. What we give you is an upper limit, right? The air, where we release and how that air moves, whatever is in the air, the PFTs are going to go where the air goes. So if you have some compound in there that's chlorine or whatever that was going to react away or going to stick to surfaces, the PFTs will give you the maximum amount that will be seen. So there'll be, maybe there's gravitational settings, something will happen that'll be less than that. But there's ways you can do that those rates are pretty well known. So if you get the PFT number, you can say, okay, in this amount of time, we're going to get this amount of gravitational settling or this amount of stickiness or whatever. And I don't think that density in terms of gas phase is an issue, because the turbulence in the atmosphere mixes well enough so that if you release, except for maybe some really extreme meteorological cases, that if you release some gas, the density isn't really an issue. Yes? Yeah, that's kind of what we did with the power plants, right? We actually took and injected into their effluent stream, and we could say that, if we know what the ratio of the SO2 is to the tracer, and we can do that. In some cases, we've actually been able to adjust the release rate to the SO2 monitors. They have a analog signal that comes out that tell you how much SO2 is, and we've been able to take the tracer release rate and increase it or decrease it as the SO2 and the stacked gases increase or decrease. So we can say when we see this amount of tracer at Big Bend National Park that then there ought to be this amount of SO2, at least as an upper limit that's from Big Brown that we're seeing there. John. Oh, okay. Yeah. There's actually some people that have proposed and have done it where they take and put tracers on money so that they can, after the money goes, they can, I think the hope was, and some people claiming that they've done it, is that the money's paid for ransom. If you put PFTs on it, then you can go around and sniff around with the truck and find out where the money's gone. Okay, I'm not real confident that that's a... What kind of hammer would that be? Pardon me? What kind of hammer would that be? It would be a mafia hammer. Work for money laundering? We've tried. Yes? We have to be careful, and there's some times where when we get really high levels of material, we throw it away. We throw these tubes away. What we do is we actually take the charcoal out of them, scrub them up, and then reload them with brand new activated charcoal. No, because the samples that we get are relatively low, but we've been shut down, and while we go and bake out stuff and try to figure out where the problem is, and sometimes that's when we get Russ to come in and wave his hands over everything. Great.
Do you have any other detection schemes or instruments you use to detect PFTs?
We have just developed one, which is a laser cavity ring down spectrometer that works, but the sensitivity isn't as good. It's in the part per billion level. Okay, but that has some specific applications that we can use it for. And the other thing that is possible that we haven't explored is using mass spec instead of an electron capture detector. Doesn't have quite the same sensitivity, but it eliminates some of the cleanup problems. Yeah. Yeah? Yeah, there's some variation, but it's not much. You know, it's fractional percentage difference, and there's some variation in isomeric composition, and that's definitely an issue, and we're trying to address that now. We have a system that we're developing to actually take the liquid PFT and get a good idea what the composition is. I think everybody's ready for wine. Thank you very much.